Let's continue with mRNA Day today. Before we welcome our next presenter, who is Daniel Anderson from MIT, just time for another quick poll and fun fact.
Hello, my name is Daniel Anderson, and I'd like to talk about our work on nucleic acid delivery systems for RNA therapy and genome editing. So first, I think there's just no question now that the intracellular delivery of nucleic acids is going to revolutionize medicine. And I think it's also very clear that the real challenge here is how do we deliver these types of therapies and molecules inside of cells in vivo in a safe and effective manner. And what's interesting today is that, now, in fact, most of us in America have already received an RNA nanoparticle in the form of the messenger RNA vaccine for COVID. But, what I think is less well known is that these nanoparticles didn't really appear out of thin air. In fact, they were a result of decades of work, and in particular on important discoveries and advances related to the delivery of small interfering RNA, in particular to the liver. And so what I want to do is give a little bit of a history lesson and a refresher on small siRNAs. So siRNA, the Nobel Prize in 2006, was awarded for the discovery and characterization of RNA interference. And this is a fundamental pathway in all of our cells whereby double-stranded RNA is cleaved into fragments of around 20 to 22 base pairs. These then interact with an endogenous enzyme system, the RISC complex. And once loaded, this RISC complex can catalytically seek and destroy complementary messenger RNA, ultimately leading to silencing of that gene. Now, it wasn't long after the discovery and characterization of this pathway that scientists and investors realized that this is a really potentially powerful way to address genes therapeutically. So if you could just get a small RNA inside of cells, it could interact with this endogenous enzyme system and essentially turn off any gene we want. And my colleague Phil Sharp has coined the term modular pharmacology for this vision that just by changing the sequence of a small RNA, you may be able to turn off any gene target that you want. And so the power of this is really incredible, but the fundamental challenge is delivery. How do we think about delivering RNAs, and in particular small RNAs, inside of cells? So when we conceptualize the challenges that we're faced with, it actually is quite a significant barrier to delivery, or series of barriers. First, we envision creating a nanoparticle and condensing this RNA into a payload system that's going to deliver it in the body. These nanoparticles, once injected, for example, into the blood, need to travel through the bloodstream. They need to avoid non-target tissue. They need to ultimately reach the destination we're trying to treat, extravasate from the bloodstream. Once out of the bloodstream, they need to enter this cells that we're trying to treat. This is typically through a process of endocytosis. However, once endocytosed, these RNA nanoparticles are trapped in an endosome and need to escape in order to release their RNA in the cytoplasm. And so it's really a complicated multi-step process, but there's been progress. And where we've first seen progress is that there are organs that are natural destinations for injected nanoparticles. So as you might expect, these organs that are constantly filtering the blood, organs like the liver, the spleen, and to a certain extent the bone marrow and kidneys, are often destinations for nanoparticles of many different chemistries. However, accumulation of nanoparticles in an organ is not the same as safe and effective delivery, but it really is in some ways the first step. And so, in fact, the first important advances in RNA therapy really were focused on these organs, and in particular, the liver. However, as I mentioned, getting a nanoparticle to the liver is not the end of the story. It's really just the beginning. RNA is a large molecule. It's highly charged. It doesn't naturally cross cellular membranes. It's prone to nuclease degradation, and it can even induce immune responses through pattern recognition receptors and TLR pathways. These can ultimately cause non-productive immune stimulation and reduced activity and toxicity for the patients. However, we've seen progress, and these are, in my mind, the three keys to getting RNA to work as a drug. First, of course, is sequence selection for a given gene, whether it's using a small RNA or a messenger RNA. There are many different varieties of sequence that you might envision using. So a lot has been learned about how to optimally identify through algorithms the sequences that are most potent for siRNA interference. Secondly, chemical modification, enormous progress over the decades in creating drug-like properties with RNA, starting back in the antisense days, but now, of course, an important part of the siRNA and messenger RNA delivery systems. So these chemical modifications can introduce stability, they can affect the potency and specificity, and they can even facilitate cellular uptake. And then finally, encapsulation, which is where I'm going to spend most of the talk. So the idea is to basically create a nanoparticle that can deliver RNA to its target tissue. And this is particularly important, of course, with long RNAs, where some of these chemical tools for delivery are not as accessible. So a fundamental question, if we're going to build a nanoparticle for RNA delivery, is what materials are we going to use? Now, I first wanted to start off by providing a recipe of sorts of your canonical RNA-lipid nanoparticle. So if you look at the majority of the work today, and there is, of course, a lot of variety, but the common lipid nanoparticle contains four components. First and most important is the amine or ionizable lipid. I've shown the structure of one lipid or lipid-like material that we developed. So this plays an important role both in interacting with the negatively charged phosphates of RNA to facilitate complexation and condensation, but it also plays a key role in organ tropism through interacting with various blood proteins. It also plays a key role in facilitating endosomal escape through interacting with the endosomal membrane as the pH changes during endocytosis. So the amine-containing lipid has been a real focus for many of us in the field because of its key role in facilitating delivery. However, in addition to the amine lipid, there are other important lipids, in particular the polyethylene glycol lipid, or a very sort of hydrophilic hydrophobic combination lipid. So these serve the role of both providing colloidal stability and also facilitating circulation time by providing resistance to protein absorption on the surface of the nanoparticle. And in addition to these, there are often helper lipids, in particular cholesterol and phospholipids such as DSPC. And so through controlled mixing, these can be combined with siRNA to create nanoparticles on the order of less than 100 nanometers reproducibly. And so when we first started this work, now dating over 15 years ago for me, but the field certainly had been looking at DNA delivery for many decades before that. But when I first started, I was, of course, naturally inspired by all of the important cationic lipid development work that had been done, in particular around DNA delivery and antisense delivery. However, when I first looked at these structures, I thought the chemical diversity wasn't as broad as it could have been. So, for example, DOTMA in the upper left here, this is a lipid from the 1980s. DLN-DMA is a lipid actually published by Protiva, an important Nature paper around 2006, really only different by a few atoms. And that's not to take away from the importance of this DLN-DMA lipid, but rather just to say at that time, the chemical diversity was not as broad as it could be. And so the first question really was a chemistry question. How do we create relevant structures quickly and evaluate those with potential for delivery? And so this is just one example chemistry that we've used. This is a Michael type addition, which is a one-step reaction between an acrylate and amine, very simple reaction where you can just add heat in a stir bar and end up with structures as I've shown in the upper right. So if you compare this structure to an old lipid, the DOTAP, you'll see a lot of similarities. They both have tails, they both have esters, and they have amines. However, this chemistry that we developed allowed us to create literally thousands of different molecules that we went on to create and evaluate in various delivery settings to look for functional delivery formulations. And so I want to provide a brief history of some of the early work. Of course, for those of you in the field, recognize that one of the key early gene targets for small RNAs was the gene transthyretin, because of transthyretin-mediated amyloidosis, which is a genetic disease of the liver in particular, where the liver creates this protein that then causes toxicity throughout the body. So we were interested in creating siRNA delivery systems targeting this gene. And so to make this formulation, we include the TTR-siRNA. We have our 4 lipid components, in particular an amine containing lipid or lipid-like material. We create our formulation, we inject a primate, and then we look at expression of TTR in the liver. And so this is data actually dating far before 2010. Working with Alnylam, we showed that we could get very potent and specific silencing of TTR after a single injection in non-human primates. And the reason I mention this is I think one of the things that was surprising is actually how many different formulations could do this. In fact, in our lab, we had identified 3 amine-containing lipids that could facilitate liver delivery in non-human primates. And at the same time, companies and other labs had developed important formulations. In 2014, Alnylam reported on their first human study where they showed these types of siRNA nanoparticles could silence TTR in ATTR patients. And this is their phase 2 data where you're looking at TTR levels after a single injection. In humans, you're getting 80% silencing that's maintained. And then ultimately, in 2018, this became the first FDA-approved RNA nanoparticle, really an important milestone for the field, because not only did it show these types of formulations really had potential as therapeutics, but also that, it was really the tip of the iceberg. It's not just TTR that can be addressed by these technologies. In fact, we and others believe there are broad areas of the body with different types of RNA therapy that can be addressed using different types of nanoparticle RNA formulations. And importantly, I think if you look carefully at these COVID vaccines, you immediately see some of the history connecting the dots between the formulation from Onpattro. As shown here, it has 4 lipid components. And if you look at the Moderna COVID vaccine, for example, you'll notice that 3 of those 4 are in fact exactly the same. The 4th aminolipid, they developed their own proprietary lipid, but you'll just structurally, you can notice some of the similarities. And of course, Pfizer-BioNTech also certain similarities in the RNA vaccine. And that's not to take away from the innovation that was required to make these mRNA vaccines. But again, I think there is this misperception that the COVID vaccines appeared out of thin air, when in fact they really reflect decades of work on RNA delivery in nanomedicine. Moving forward, I want to sort of introduce some other technology that we've been working on, which is nanoparticle-mediated RNA delivery using polymers. And so I think there may be a misperception that RNA delivery systems are good for vaccines and maybe hepatocytes and that's it. But in fact, we believe that nanoparticles can address other parts of the body. And in fact, they don't need to be made out of lipids. And this is an example of a composition based on biodegradable polymers. And so we've been making RNA nanoparticles from degradable polymers. Unlike the lipid nanoparticles, sometimes we use additional helper lipids and sometimes we don't. But in all cases, instead of an amine-containing lipid, we have an amine-containing polymer, and it has to be biodegradable as well, or at least is preferably degradable to facilitate clearance. And so here's an experiment where we developed a polymer RNA nanoparticle with 5 different siRNAs targeting 5 different genes, in this case in the endothelium. And when we create this nanoparticle, it's very small, in the order of 35 to 55 nanometers. When we inject a mouse, we look at the lung, we see actually silencing of all 5 of these gene targets: Ty1, Ty2, VEGFR2, VE, cadherin, and ICAM2. And what you're looking at here is silencing of these 5 genes as decreasing a total dose of RNA. So even at the lowest dose of total RNA tested, we're getting good silencing of all 5 of these gene targets. And this is exciting both because it shows that polymer nanoparticles can also silence in vivo. It shows that it's not just the liver where we can get silencing. In fact, it is other important organs like the endothelium. And then finally, it shows that these systems can be multiplexed. And so I work in a cancer center where there are diseases that are complex, and perhaps silencing one gene isn't going to be enough to provide the therapy. Here's an example where we in fact silence 5 genes, and in fact, working with others, we've been able to silence as many as 20 genes with a single injection in vivo. I'd also mention that this early work was in rodents, but we went on to publish a paper in 2018 showing this formulation could also silence in non-human primate lung. So finally, getting back to messenger RNA, I don't think I need to define messenger RNA for this audience, but in my mind, in some ways, messenger RNA is the other half of modular pharmacology, where siRNA is turning genes off. Here, messenger RNA is turning genes on and leading to the production of new proteins. And again, the real question is delivery. The central dogma of molecular biology is that DNA leads to RNA leads to protein. But the question is, how do you get mRNA inside of cells in a safe and effective manner? And when we think about the delivery challenges, well, siRNA is in fact quite hard, or at least we imagine quite hard to deliver. Messenger RNA seems to be much harder. First, it's largely single-stranded, so that means it's more susceptible to nucleases. There's a limited set of chemical tools available for synthesis of these molecules, and in fact, they're very, very big relative to small RNAs and conventional drugs. However, of course, there's been progress, and I want to give a couple brief examples. Here's an example of us comparing 2 types of formulations. One, a lipid formulation that's designed to target the liver, and second, a polymer formulation that's designed to target the lung endothelium. And so when we use a messenger RNA encoding luciferase and inject a rodent with these nanoparticles, what we can see is that it's possible to build nanoparticles that will access different parts of the body by altering their chemistry. And so here, what you're looking at is in the lungs, the polymer nanoparticles are giving us very good expression in the lung. However, the lipid nanoparticle is giving us basically no expression in the lung. In contrast, the polymer formulation is giving us no expression in the liver, whereas the liver formulation is giving us very nice expression. I'm showing that again, changing the chemistry of a particle can change where it goes in the body. Now, my colleague, former student, actually, Daniel Siegward, published a very nice paper last year showing that it's possible to retarget lipid nanoparticles to different organs by adding additional helper lipids. And so in particular, he was looking at the addition of cationic and anionic helper lipids. And by adding cationic lipids, he could take a formulation that was largely going to the liver and then now get retargeting so it's basically completely going to the lung. And so this is a nice example. It's not just that polymer nanoparticles can get us to the lung. In fact, lipids can also access the lung endothelium. I want to describe a new library of lipids. We call it the IR series. And these are biodegradable lipids that are endothelial-specific. However, we are not adding any additional helper lipids. So just by changing the chemistry of the lipid, we're able to get retargeting of this lipid formulation to the lung endothelium. And in fact, when we compare this to, for example, the C12-200 sort formulation, we're getting several-fold better delivery at the same dose. And in particular, we see the targeting is very, very specific. So we're not getting low-level expression in organs like the spleen, but it really is centrally focused on the lung. One helpful analysis that we've done leverages an assay system we published in 2018, where we use a lock-stop-locks TD tomato model, a mouse, where we deliver Cre recombinase messenger RNA. And upon successful delivery, you will get permanent removal of the stop site to allow for expression of TD tomato. And so this is a nice way of looking at what percentage of the cells are getting functional delivery. And so when we do this in this mouse, in fact, basically get 80% of the endothelium of the lung being converted. So we're getting very high levels in the animals in vivo. And importantly, it's also basically specific to the endothelium. We see no conversion of epithelial or immune cells. And I'd also mention, again, compared to this C12-200 DOTAP formulation, we are also getting a higher level of conversion. However, there are, of course, situations where we don't want to access the endothelium, we want to access the epithelium. And so here's an example where we can actually create nanoparticles with messenger RNA and nebulize them. And so in this experiment, we take polymer messenger RNA nanoparticles, subject them to a vibrating mesh where we make water droplets that have the nanoparticles contained in them. These can then be flowed into a cage with mice so they can breathe them in. And when we do this, we get very nice expression to the lung epithelium. It's a uniform distribution across all 5 lobes of the lung. And importantly, this expression really is limited to the lung. We see no expression in the kidneys, liver, or spleen. So finally, now I want to switch to thinking about how we might use this to repair your DNA while you're still using it. And this was actually the dream that brought me to MIT actually back in 1999 was, is it possible to create a nanoparticle, say you have a liver disease, which is why I've drawn this in green, that would travel through the body, reach the liver, enter those liver cells, deliver a payload inside of those liver cells that could then specifically correct the gene defect that would lead to permanent restoration of function in your gene target. And so again, it sort of really sounded like science fiction, but now we have really exciting evidence that this is possible, not just in animals, but now also in humans. Now, the trick to getting genome editing to work, and I will sort of give a caveat because every day we seem to have new exciting ways to advance genome editing with exciting new technologies. But I would say, in the beginning, really the key is to create DNA repair events that can ultimately provide therapy. So we want to take advantage of our own DNA repair pathways to create a therapeutic editing event. And so we've been focused on the CRISPR-Cas9 system. This is an enzymatic system that will cause a double-stranded break as guided by the guide RNA. And so in order to get this double-stranded break, we need to deliver the enzyme system protein, but also the guide RNA to direct cleavage. And so I'm only going to talk about one form of editing, which is indel formation. In this setting, we deliver the Cas9 with guide. It creates a double-stranded break. And now non-homologous end joining machinery in our cells will then close that up by gluing the ends together. However, if it's a perfectly repaired substrate, it will be cut again. And this process continues until there are frequent chances where you can get deletions or insertions, ultimately leading to premature stop codons in the gene. So it's a way to permanently turn off genes. And to do this, again, we need to deliver Cas9, which we've used the messenger RNA coded Cas9. But the question at the time was, how might we deliver the guide RNA? And so we reasoned that, in fact, one way to facilitate delivery would be to take advantage of all the important chemical modifications that have been developed for antisense and siRNA. And this is a partial list of a partial list. In fact, it's really incredible how much chemistry has been done here. But in particular, chemistries like the 2'-O-methyl, 2'-fluoro, and the phosphorothioate have sort of risen above as being particularly helpful in facilitating siRNA and antisense delivery. So there are many chemical modifications that have been used, but the question really, when we wanted to think about modifying the guide RNA, was how can we modify it and identify which ones are the most helpful? The fundamental problem is it's around 100 nucleotides. And if you imagine all the possible chemical modifications to backbone and bases, and at each site, it really becomes an intractable problem. And so what we decided to do was, instead of, combinatorially testing every single variety, we looked carefully at the crystal structure that had been published by Jennifer Doudna and Feng Zhang. And so we were able to make predictions about which regions of the guide RNA we could chemically modify and still allow for function. And with this, we came up with the enhanced guide RNA, where in particular we see these loops tend to be chemically modified. We identified several reasons where we didn't modify. And so to evaluate the in vivo performance, we targeted a gene called PCSK9. PCSK9 is a gene involved in recycling of the LDL receptor. There are antibodies that you can take actually that will reduce your cholesterol by over 50%. But these antibodies need to be taken for the rest of the patient's life. And so we thought perhaps we could build a nanoparticle that might permanently lower cholesterol. And so to look at this, we created a nanoparticle containing the messenger RNA with the Cas9, 2 chemically modified guides targeting PCSK9. We create our formulation, inject a mouse, goes to the liver, and then we look at the liver genome and also blood cholesterol. And what we found after a single injection is we could get 80% knockdown of, we could basically hit 80% of all of the genes in the liver. And following this, in fact, the serum PCSK9 went from detectable to undetectable. And while cholesterol metabolism is a little bit different in rodents, it led to a 35% reduction in total cholesterol. And so this was an important early demonstration that it was possible to build just a nanoparticle, LNP, that could dramatically knock out a gene in vivo. And we published this back in 2017. More recently, a company called Intellia has advanced their RNA-Lipid nanoparticle formulation with a messenger RNA and a chemically modified guide into the clinic. And they've been looking at hitting the same gene that this first siRNA drug hit, the TTR gene. And so they released this data just this June. And at the highest dose tested, you're looking at serum TTR levels. In humans, you're getting over 80% reduction, which is in the therapeutic range. So a very exciting demonstration. And they also included a variety of studies looking at primate delivery and other aspects of this. But the bottom line take home is that this is in-human proof that lipid nanoparticles can be used to facilitate in vivo genome editing. So finally, I want to spend my last few minutes talking about a new form of RNA, which is circles. I think there are many types of RNAs being evaluated, but we think in particular circular RNA has potential as a unique platform. And so first, circular RNA is found in nature and is generated through multiple mechanisms, typically through exon backsplicing. So if you look in a variety of tissues in the body, you will find circles in your body, in particular in the liver and the heart and others. However, when you look at how circles can express proteins, they really are fundamentally different. And so, your canonical messenger RNA has a cap and a tail. Circles have neither of those things. And so, of course, the factors involved in translation are very different. However, because they don't have ends or caps, they're both easier to make and also resistant to nucleases, which can lead to more durable expression. And so we've created and described the first easy ways to create circles through a self-splicing system, and also methods for purification to make them immunosilent. So we use no chemically modified nucleotides. All of the circles are full length, so we only have full length transcripts, and these have high payload capacities beyond 10 kilobases. Importantly, these are immunosilent in vivo, so I don't have time to go into this particular formulation, but when we look at in vivo delivered siRNA, we see no evidence of silencing. This is also true, no evidence of immune stimulation is also true in vitro. Importantly, these circles provide a more durable expression. First, this is in vitro data, and so you're looking at expression in days in HeLa cells compared to both modified messenger RNA and unmodified linear RNA. And this is work we published a couple years back. More recently, and this is some unpublished data from a company we formed called Orna, we've been able to identify RNA constructs that are able to express at very high levels in human T cells. And so what you're looking at is a variety of constructs in comparison to messenger RNA, which is what's shown here in black, where you see very high levels of expression and more durable expression in human T cells. And so we're excited by the potential of these types of constructs to provide for in vivo CAR T therapy. And importantly, because of the ease of manufacture and the lack of the need for the capping reagents and et cetera, these are much simpler to make, and the overall cost of manufacture is about an order of magnitude cheaper. And so when you think about the cost of goods, we see that as a key advantage of circles, even beyond their improved performance in therapeutic setting. So finally, I need to thank many smart people I've had the great pleasure to work with. In particular, for this, I want to mention collaborators at Orna, in particular Alex Wesselhoff, who was my graduate student, one of now key scientists there. I need to thank in particular Hao Yin, Kevin Kaufman, Luke, and Idris, who are in my lab developing these new endothelial delivery systems. And with that, I want to thank you for your attention. I'm happy to take any questions. Thank you.